Okay, this class is on drug discovery. So the drug discovery is a multidisciplinary research. It comprises several steps and several fields of expertise, starting from target selection, lead discovery, medicinal chemistry. So first, we need to start from having a cellular target or genetic targets, and then combine the field of genomics, proteomics, bioinformatics. This field will lead to the target, and the target will undergo combinatorial chemistry screening, which they use cocktail of ligands to find the hits or the compound that seems to have activity through high throughput screening. After the high throughput screening and they got the molecule called the hit, it will undergo structural activity study, in silico screening, chemical synthesis. So the chemical synthesis person will synthesize the molecule, and then the molecule will be tested in the test tube called in vitro study. In the in vitro study, they will test for the mechanism of action. These molecules, once they pass certain refinement, they will be called a lead candidate or lead molecule. The lead molecule that satisfy the mechanisms of actions and absorption, excretion, toxicity, and all those pharmacokinetics parameters, they will undergo in vivo study, which perform in the animal models such as mouse, rabbits, monkeys, and at the end, they will be tested in the human during the clinical trials. So there is several phases of clinical trials, and normally before the pandemic era, one drug molecule would take around 1.3 billion US dollar and around 17 years from the beginning to the end of the process. And this is the paradigm for structure-guided drug discovery. First, that would be the gene, and the gene of interest. If they got the chunk of the genes that are the target protein, they will use that as the target for the drug molecule. So they will design something which fits inside the pocket in the way of the three-dimensional jigsaw puzzles. And once the molecule fit in there, that's suitable for the size, shape, and charge, that will become a new drug. So the terms to understand the first concept of ligand-based drug discovery is the chemical space and then the descriptors. So what is the chemical space is that people thought compounds in the universe is as much as the stars in the sky. And they try to categorize those stars in the sky or the compounds universe into classes of protein targets, which are like kinases, serine proteases, metalloproteases. So these are all drug class that they thought the molecule in the universe would fit into. And they try to group them, and they thought they will be able to discover the drugs for specific classes inside the chemical space. But since the chemical space is so much like the stars in the sky, that would be impossible to synthesize all the compounds and test them all. But the drug company spend large amount of money and manpower in order to screen the chemical space. So the first concept that we will discuss is the ligand-based drug discovery. So the term molecular descriptors is that it is a way of expressing and analyzing chemical or structural information. The commonly used molecular descriptors may be physical chemical properties of a molecules, such as molecular weight or log P, or the descriptors that are derived from 2D graphs of molecules. So like a representation of graphs of molecules in numbers. Or they may be derived from the three-dimensional structures of a molecule, such as the pharmacophoric keys. There may be counts, such as hydrogen bond donors and acceptor counts, aromatic rings, rotatable bonds, molecular weights. Or there may be hydrophobicity terms, such as the logarithm of partial coefficient between, so this is called log P. And there may be topological indices that describe the size, such as if the size is large, they put large numbers. If the size is small, they put small numbers. And they try to get the idea of branch and overall shape by describing the size with the simple numbers. So these descriptors, they will be used in order to derive the values of the activity. From high school mathematics, we can know that there is the equation, linear equation, that is the simplest form called like y equal to mx plus c in the expression. So there is this one is the slope or the coefficient. And if you know the value of x, you can predict the value of y. If the activity is the activity, such as activity is small, there is certain number. If activity is large, there's certain numbers. If you know the value of x, you should be able to predict the value of y, as in whether the activity should be better or worse. And the equations that you can solve this is like linear equation y to mx equal to mx plus c, but it's more complex than that. That would be called the multiple linear regression. So in the linear regression, you can have y equal to mx plus c. Can you guess what is the m in this equation? No one can know. If you put x here, you get y here, the first y1. And if you put x equal 2, y equals 7. If you put x equal 3, y equal 10. Can any one of you guess what is the m or the c? Can you guess what is the c values? No one. So basically you have 4 here, right? And if x equal to 1, and if the next equation you have y equal to 7, x equal to 2. So if you have m equal to 3, can you guess? So that is the C number, right? So in the same way as descriptors, you have the value of y, which is the activity of the drug in the protein target. And you know various number of x as descriptors I described before, that may be the number of rings, the size parameters, the number of rotatable bonds, the log p value or whatever. So you have so many values of x, and you try to guess the activity that are desirable for your certain subjects. So by this way, if you know that x equal to 1, m equal to 3, you know the value of c, so you can guess the value of y from m is constant and c is constant. So you have so many columns of x in the real circumstance, such as in capsaicin analogs, you will have something like this table. So the y value is in log 1 over EC50. So it has to be the log scale because it's ranked from like millimolar, micromolar, nanomolar. So 10 to the power of -3, -6, -9. If you want to put them in the same graph, you have to put it in the log scale. So from the log scale, you rank from smallest to largest. And then you have many values of X, such as MR or molecular refractivity, pi, which is the hydrophobicity parameters or how far hydrophobic they are, electronic sigma constant, like here, and top size parameters. So you have so many values of X in many columns, and you have columns of Y. And you use these X's in order to train how to find the M and C values. So once M and C is constant, you want certain value of Y, such as if activity is very, very good, you will know which parameters should be put as an X. So that's the basic concept of ligand-based drug discovery. Once you know this concept in mind, you'll know, okay, they will just have so many descriptors, just whatever they are, and then they will try to guess the best activity out of the value of X. So you have Y here, have X1, X2, X3, X4, and then this series will be used to train to find the model or the equation that are the values of the coefficient and the values of the C values, for example. So example, this is capsaicin analogs. Do you know what capsaicin is? No? What plants capsaicin exists in? So they are in chili that causes spiciness in the food. So in capsaicin analogs, it will have the equation like this. So log 1 over EC50, this is the value of Y. And then there is this is molecular refractivity and pi and sigma constant and ES. So this is coefficient and X, coefficient and X, coefficient and X, and this is the intercept. So if we get the equation like this, we can design a better molecule, such as we design whether the ring should have bigger size or should have certain electronic properties. So that's the concept of descriptors. There is a field called COMFA. So the COMFA, basically, they will have molecule of the ligand or the drug compound that binds to the protein, but they don't know the target protein yet. So they basically get all the ligand and then they plot the pharmacophoric point, such as this one point here, 1 point here, 1 point here. So they find the similarities among them. And then they will designate this as the size and type of pharmacophoric keys. And then they superpose the triangles together. And if they train large amount of ligand in the same series, they will know that L at this corner, this L, this L, this L, normally should have certain electronic properties, such as should find the negative charge very often, for example, or they should find the steric interactions. So they superpose this ring, this ring, this ring, and this ring, and then they get the field out of it. So they put those pharmacophoric keys and the ligand structures into the grid like this. And at each grid points, they would have the electronic field. And then from the molecular field, they will plot the tendency of certain parts to meet certain electronic properties like this. So this is the ring, the tree ring here. And then if it has the positive charge characters very often, if it's often meet the positively charged characters, they put blue colors. If they often meet the negative charge characters, they put red colors, something like that. Or if they often meet the steric or the hydrophobicity parts, they will put the green color. So by having just the ligand alone, they can imagine the surrounding that should often be in certain properties. So this is the old days when the X-ray crystallographic structure is very expensive and very hard to solve. That's how large amount of drugs discovered in the old days. They generate a lot of these ligands, and then these series they use to generate the molecular fields as in COMFA or COMSIA, as you have covered in your essay. So that's the ligand-based drug discovery. The next concept is structure-based drug design. So structure-based drug design or rational drug design comprises several fields and several experts. The X-ray crystallography or NMR spectroscopy will be the field that used in order to discover the target enzyme structures. The target enzyme structures will be used in order to design the molecule by computational chemists. And then the computational chemist will pass that molecules on to the organic chemist to synthesize the inhibitor candidate. After that, it will undergo in vitro testing in the test tube, in vivo testing in rats, rabbits, monkey, and mouse. And then it will pass on to pharmaceutical chemistry testing until the clinical trials. So this process will repeat it so many times and take years and take a lot of manpower. But it's more promising than just guessing by the ligand alone or by trial and error to try high throughput screening, because we have the target enzyme structure and we know the size and shape of the pocket of the 3D puzzles that we will use in order to design the molecule to be specifically bind into it. So my expertise is in computational biology or bioinformatics. So the parts that I can help in this map is around from target enzyme structures to the drug design analysis. So I built a web server, algorithms, and tools to foster the collaboration between the structures from X-ray crystallography to computational chemists as a designer of the drug molecules. So this one, I think I have covered in the bioinformatics class before, but I think I need to remind you again all these concepts so that you can quickly grasp the concept of structure-based drug design from the structures. So the protein structure, starting from primary structure, which are the sequence, that 20 amino acid sequence in letters. And if they have repeating torsions, there will be the secondary structures, such as if they have spiral shape, there will be alpha helices. If they have a back and forth characters like this, going to the right and going to the left, this will be called beta sheets. And if there is a combination of many secondary structures, such as beta, alpha, beta, or alpha, alpha, this is called the motif. And once the motif have a recognizable fold, such as in clam or in sandwich or in basket or in solenoids, horseshoes, those will be called domains. And if they have functions, that will be called the tertiary structures. And if the tertiary structures have cooperativity among them, those will be called the quaternary structures, as in hemoglobin that have 4 units, for example. So this is the periodic tables. And if we put it in molecular visualization tools, we will see the sphere with radius size like this. So the rows that we often use is these rows. That is carbon in white, nitrogen in blue, oxygen in red, and sulfur in yellow. So you only need to memorize these 4 colors. And this ones, I think, is covered extensively in previous class. So shall I repeat it again, or you want me to skip this. No one.
You can repeat it again.
Okay, so glycine is the smallest unit. It will be used in the structure as in the part that require flexibility. So glycine, or the G value, the G letter in here, this one will be the smallest one with no large groups, and it allows flexibility of the loops of the protein. So if there is hairpins or something like that, at the turn, that will be the glycine, because it allows for the turns and flexibility. Alanine, valine, leucine, and isoleucine, you can see they are all gray. These ones will be the hydrophobic, small to medium size, and they are uninteresting ones. They mainly serve for the structural role. They display structural role. They are there for their size and shape. And serine and threonine, this one, serine, threonine, or tyrosine kinase, will have one red tip here. The red tip will allow them for phosphorylation. Basically, they add phosphates at this OH or hydroxyl group here. So if you see the shape with one red tips, serine, threonine, or tyrosine in the next page, but they are rings. So this one will be for post-translational modification at the hydroxyl group. If they are yellow, cysteine, it will be reducing agent. It allows for cysteine bond formation. If they have two red tips, as in aspartate and glutamate, these ones will form salt bridge with the positively charged amino acid. So they normally call, they have partial positive. They are not full positive, but they are partial positive. So this one, partial negative charge, because they are red to red make minus. So negative charge, partial negative charge of aspartate and glutamate. Aspartate and glutamate is different by one carbon. So this carbon of glutamate is extra from aspartate. There is one carbon longer, but the charge and the shapes are similar. Asparagine and glutamine with one letter N, one letter Q. These ones have one positive, one negative, so they are polar uncharged. So they have blue and red. This is polar and uncharged. So these ones, they are polar, they can dissolve in water, but there is no charge characters. And you can see lysine, arginine, histidine. This one have blues, see, lysine, arginine, histidine. They are partial positive charge, and they form salt bridge with the partial negative charge. So this is positive from salt bridge with the negative. They are also allows for epigenetic, they are epigenetic related protein, because they have these positive charge characters. And for tryptic digestion, as in proteomics, they also looks for positive charge molecule in order to recognize the proteolytic cleavage site, as in lysine or arginine. And proline, the proline introduces king in the helix, because they roll back from the side chain into the main chain into the cyclic ring formation. And this one, if the helix go in the spring, like longer in a long spring, if they find the proline, it will introduces king in the helix, because it has turning characters. And phenylalanine, tyrosine, and tryptophans, these are aromatic residues. So they have a 6-membered ring, and this electron can be delocalized. They can form pi-pi stacking interaction, that is the pi electron and pi electron stacking, like in the wheel, the two wheel that stack together. So this one, as I say, if they have one red tips, it can be phosphorylated or allows for post-translational modification. So these ring amino acids will allow for chymotrypsin recognition, as in they often recognize large hydrophobic group, basically mean large aromatic rings. So they use for molecular recognition of chymotrypsin, for example. And the side chain representation. So if we look into the helix and look at the glutamate and histidine side chains, you can see that if the glutamate with two red tips and histidine with 5-membered ring, if we draw it in sketching, like in chemists, they normally use for sketching chemical synthesis, they will draw it as line representation with colors like this. But a more accurate pictures that they use in the paper, in scientific paper, they will normally use ball and stick representation in order to see the sphere with larger size than the bond size. So the radii of the sphere larger than bond, it will be called ball and stick. And if you are crystallographers, they will draw the sphere of the atoms and the radii of the atom and radii of the bond into the same size. So it will look like this and called the stick representation, because the radii of the sphere and radii of the bonds are similar and they look like this. So this is what used among crystallographers. And if you are drug researchers, normally you want to get the true size of the atom, as in how large oxygen or nitrogen atoms are, and you normally want to represent it with van der Waals surface of each atoms. So that would be called the CPK space fill or van der Waals surface representation, that represent the true van der Waals radii shape of the atoms. So this is CPK space fill. So this one's is easy. So there is C alpha trace, ribbon, schematic, van der Waals surface. So previously, this is just one helix. If you have so many helix like this, and this is the protein instead of just one alpha helix representation. So for protein representation, if you use C alpha trace and use one color per unit, you will have like this one, 6 structures. And if you have ribbons for all of them in the cyan, in this one as beta sheet in cyan, red color for the helix, you will have the ribbon. And schematic have direction, and van der Waals surface have, normally they color it by charge properties. You can see the positive charge in blue, negative charge in red. So let's see in details. So if you dissect only one helix here, you can see that if you omit all the side chains, it will be called a C alpha trace. So you discarded all the amino acid side chains. So this is the C alpha trace, look like noodles, like yellow noodles shape. For solid ribbon, you can see that the helix just basically drawn from having the main chain and then have the size of the main chain in so many lines and rendering until it becomes the ribbon. So they have the thicker line along the alpha carbon, and then once they trace all the alpha carbons and omits all the side chains, it will be these structures called the helix structures. Or if you want to see the turn or the beta-pleated sheet, it just go parallel to one another like this. And the turn normally colored in green and the strand colored in cyan. So this is the schematic representation, which similar to ribbon, but in contrast, it has direction. So this one, it just traced along the main chain, right? But if becomes schematic, they will link the spiral shape into one rocket, like in Pratat. So they will have direction, like this one, glutamate histidine go up to N terminus and N at C terminus. So start from N terminus, the nitrogen atom is on one side at the beginning and go towards the C terminus of the protein. And if there is the beta pleated sheet, it also goes from N terminus to C terminus. So it will have the arrow shape, and each sheet will have the N terminus to C terminus direction of the sheet in arrows. For van der Waals surface, you can see that each atom will have its own van der Waals radii. And if they combine the representation with the color that is putting positive in blue, negative in red, they will see the partial charge characters. And if they want to design molecules, they will match negative charge of the ligand to positive charge of the pocket, for example. So that's how they design the molecule. So normally they use van der Waals surface to see the true shape inside the pockets. So what makes good drug target? Almost all drug targets are protein. DNA is targeted by some anti-cancer drugs, but not very often. It should have known mechanism of action. And so specificity is also very essential. There should be no other similar molecules that do the same thing inside the human body to avoid side effects. And it should be druggable. That means the binding size should have a size and type that can be targeted by small and tractable molecules. So the pocket should be friendly enough for the molecule, the small molecules, to sit inside the cleft of the protein. And for target identification, the researcher will ask what protein targets are good for this disease. Like in COVID-19, right? We would look for main protease as the drug target because it cleaves proteins that are essential for the viral propagation. For target validation, they will ask whether the chosen target are druggable. And for lead identification, they would ask which small molecules should have the right activity at the chosen target. And for lead optimization, they will go to the variants that should have right pharmacokinetic properties and non-toxic. Pharmacokinetic properties, as I discussed earlier, will be the absorption, excretion, metabolism, and so on. And toxicity, something like ADME-tox, something like that. So let's see the simple models that shows the human immunodeficiency virus and how to design the anti-HIV compound. This animation produced by Janet Iwasa. At that time, she was at Harvard Medical School, and now she is at University of Utah. So this is human immunodeficiency virus, have envelope protein and viral envelope. And inside there is capsid, matrix protein, and accessory protein. Inside there is the RNA inside the nucleocapsid. And this is the size of HIV in comparison with the T cells. you can see that HIV is very small, and this is human T cells. Upon infection, HIV will try to get inside the T cells by rolling around the surface and then trying to catch the surface with its receptors. And once the receptors binds, it will try to punch a hole with a red helix to push its genetic materials inside the T cells. So this process, it will push helix inside and punch a hole, and then these red helices will allow for the two membranes to fuse to one another. And now it can inject its genetic material inside, and then it getting inside the T cell and infection occurs. So a possible strategy, previously we talked about small molecule. Now let's see the peptidic ligand concept. When they try to design the peptide that mimic the blue molecule here, so the green molecule will act by binding this red helix during the hole punching process, but it doesn't allow the two membrane to fuse. So that's one strategy to avoid infection, because once the membrane cannot fuse, it cannot infect the T cells. So that's another class of drug. So previously we have small molecules. This is peptidic ligand. And another process is HIV viral egress. So this animation also generated using real PDB files. During viral egress, when they try to exit out and infect other cells, it will recruit several genetic materials, and then it trying to gather all these associated molecules and try to exit. So when we look for target protein in order to design drugs, something like this also important, such as these cyan molecules that would allow the cells, the HIV, the newly generated cell to exit from the previous cells. So it will go like that. So if we stop the process by blocking this cyan molecule process, this is also the drug target that we look for. So once you understand this into pictorial representation, you can get a little clearer concept of how to design drugs and what are good targets. So the targets that people study a lot, normally for the cancer drugs, they will study the protein kinases. Because the clef is so similar, all protein kinases all binds to ATP. And these ATP pockets are similar amongst various classes of kinase. So you can see that these cells also very important for cell cycle control and cell growth, cell metabolism, cell differentiation. If we can block the cleft inside the protein kinase with the suitable drugs that mimic the structure of ATP, we can shut off the cell cycle and lead to the curation of the cancer. So the wonder drug is the Gleevec that are very successfully used for treating chronic myeloid leukemia. So inside the cleft, the interaction that we should be considered very clearly are these molecular interaction. So there is molecular forces that affects the binding affinity, such as the covalent bond that share electron pairs between ligand and target atoms. So they share electrons pairs, and then The example of covalent bond is this one. So this is the structure of cytochrome C, as I have mentioned in the bioinformatics class. You can see that inside there is the heme molecule, and the heme binds to the pocket of cytochrome C using covalent bond. It has a permanent bond inside due to the sharing of electron pairs. So another interactions such as van der Waals interactions, which are causing by transient dipoles. So this van der Waals interaction caused by transient fluctuation of electrons inside. And there is ionic interaction, which is the attraction between cation and anion. So as I told you, there is partial positive charge and partial negative charge. And this red binds to blue like this. So this is ionic interaction or salt bridge. And the molecular forces that also affects binding affinity that you should consider clearly, such as the hydrophobic interaction, which is the aggregation of nonpolar molecules that cannot hydrogen bond with water molecule. So this one, I often make a joke, like this one is the concept of Lakontai. So this is the molecule called NAM, and NAM has a friend who doesn't like her and have a bad girl who hates her as well. So her friend who doesn't like her and also a bad girl in the movie, if they hate NAM, they become the friend to one another. So they hate the same thing, that's fine, they form clump together. They gather together because they hate water. So this is the hydrophobic interaction. They try to exclude themselves out of water in order to be with the one that hates the same thing. And this is hydrogen bond, is the interaction between hydrogen atom with a partially positively charged and another atom with unpaired electrons. So this one should be covered in chemistry class, but just try to get concept first, that is, this is the hydrogen bond. And if you want to do a degree in drug design, you understand further about hydrogen bond, which is very, very crucial for the drug designing. So this is molecular docking. It's based on lock and key concept of pharmacology. The aim was to find molecule with suitable conformation by using the thermodynamics free energy terms to filter the possible pharmacophores of the ligand. So it used a little bit of simulation in combination with the free energy calculation. So it tried to find the lowest energy conformation of the ligand, the shape that are very stable, have low energy, in order to fit inside the pocket of protein in the same way as the lock and key. So let's see the animation. So this is the schematic representation. You can see arrow have shapes and the line representation. This is the van der Waals representation, and the molecule of the ligand in green is optimized in order to find the most suitable conformation. Once the molecule has suitable conformation, it will be calculated to find the lowest energy conformation. So you can see the energy getting more minus means lower and lower. And after the free energy is lowest, it becomes the molecules that will be used as the lead molecule. เหลือ2 คน อีก 2 คนยังไม่มา so far you understand? Do you understand the lecture? Yes, no? Yes? Is it too fast, too slow, too fast?
I think it's quite fast ครับอาจารย์
It's quite fast. Okay. So, but I hope that by doing an essay before, you can grasp the concept quickly without too much burden on the new concept. So that's why I request you to write the essay one more time. So, do you want me to explain more clearly about which concept? No? Yes? Is there any part that you do not understand before I continue? Yes, no?
Could you like summarize about the concept y equals to mx plus c, about which one that so I mean c, and we have to find c and x, right, to identify the value of y in terms of reporting.
See, you can gain it. That's the field of machine learning, the basic of the most basic form of AI. So what the machine learning does is it has tremendous amount of X. And then if they have so many value of X and so many value of Y pre-existing, so they know Y and X before, such as if they have 20Y and 20X, they can guess C and they can guess the coefficient. So guess C and coefficient, which is constant. And after that, they use C and M as the model equation. And then they want to name Y1 and then they guess what X should be put. So let me introduce the slides. So in machine learning, you have this series. And then this one is X1, X2, X3, X4. And they have Y like this. So with this one, for example, if you already know this row up to I, you have X and Y. You just look at MI as 1X and Y. So you know this row up to I here. And you have these values, right? So you know the column of Y and column of X. And you can guess by this one, if you put M equal to 3, C equal to 1, for example. So C equal to 1 times 3 becomes Y equal to 4. And M equal to 3 times 2 plus 1, Y equal to 7. If M equal to 3 times 3 plus 1 equal to 10. So by this one, you know the black value, M and C. And then you train it already. This is training set. You stop there. Then you want Y, which is the activity, equal to 9. You have 9 here. And you know M and C from the training. And you can guess what X should be. Right? And X, if it's equal to charge, if it's equal to number of rings, if it's equal to the size, you can guess what X can be by training large amount of X and Y to get M and C as the model first. And then you can pick one Y and then you guess what X can be. Just concentrate on here. You have the concentration that kills the enzyme by a half. So that's the activity, the concentration that once the enzyme got is the concentration that kills the enzyme. And then it has C-intercept. And then coefficient that you get from machine learning, from training the models. So you have these trained value and you have X. So if you have this equation already and you want log 1 over EC50 equal to 9, you can guess X from here. So all these, you don't have to calculate by paper and pencil. It has the machine, basically just like Excel spreadsheet. So you put this value in Excel. And if you understand the concept, you choose this as X, you choose this as Y, it will throw the value of M and C for you. And then you can say, okay, if you want Y equal to this, and then it gets all the X's.
Thank you.
Okay. Any more question? So we need to understand the previous thing first in order to understand the later part of the lecture. Any more question? No? Okay, let's move on. The next concept is the novel ligand design. So at the beginning, we have the shape of the pocket. Like here we have semicircle, have triangle, part of the triangle and part of the squares. So we have shape of the ligand. No, this is shape of the pocket. And we have chart characters in colors. So what we want to do is we want to design something to catch with the clef that match with the shape and charge. So shape and colors. And so in the novel ligand design, that means generated anew. So we want to find the shape of the ligand that fit with this clef. And then we link them together in order to find a new molecule that binds to this cleft. So this is the novel ligand design concept. And this is the real case. So this is the ligand. You can see the blue atoms here. That's 2 nitrogen. And these rings, it's fine to bind aspartate. So after the curation of the, I mean the database person found this and then they try to collect the data, they will capture this as aspartate molecules normally binds to this molecule. So they cut this molecule and have this and put it in the dictionary. So this molecule binds to aspartate before. And then they put it here. And from statistics, they put this aspartate here. Aspartate and glutamate has the same shape. And it's found that in aspartate, 72 times it binds to this molecule. So this molecule binds to aspartate 72 times. And going back to here, aspartate here, for example, and that 72 times have red circle. And so this dictionary will be inside the database or inside the mind of the medicinal chemist. So they knows that if they see aspartate, which option they would put in order to fit, and then they link them up in order to find the whole. So this is the de novo ligand design. And next concept, this is completely new one called fragment-based drug discovery. So this is Professor Tom Brundle.
There and there with the same library.
So he have shapes like this as well. Shape semicircle, triangle, square. And then what he did was fragment-based drug discovery by gradually growing the ligand until fill up the whole pocket. And each step require X-ray crystallography. So he solve structure each time and then grow it each time and X-ray crystallographic structure of it. So you can see in the video.
But we'd have to detect them with biophysical methods or X-ray methods to see where they went. And I thought in 1997-8, what we would do would be to cross-link them all. In fact, it turned out that the best way was to grow them. And so then what happened is we proposed to venture capital that we would have an experimental system, we'd have known structures, and we would screen those with a very small library using high throughput biophysical and X-ray methods. And sooner or later we would get one that binds, although I sit here sometimes wondering whether I will, and there you are. That's a fragment bound. You wouldn't get it in a biological activity, but you can spot it and define it using the sort of methods, X-ray methods, NMR, and many other methods. And then we get chemists in to elaborate it. And so that's the general idea. And then I went to two students of mine. Well, I should say this one, Duangrudee, was really, as much as anybody, the driver of this. And he was in Glaxo Wellcome at the time. Here's Chris Abel, who's another academic, professor of chemistry. And the three of us got together and eventually persuaded yet another one of my students to give us $500,000. And what we did is we had two people in my lab and one in this guy's lab, and we tested out the idea. We could see these small fragments and then begin to do something with them. And within 6 months and totally finished within a year, we showed that we could. We then advertised ourselves. We raised $20 million. And then we recreated our academic lab, the chemistry lab and the biology lab, up in the science park, a couple of miles away from my academic lab. And then we built a big building, of course, and we had all sorts of collaborations, but they were all based on getting a target. We have around 30 chemists, by the way, and we make the chemists sit round the computer and say, well, look, this fragment binds, you can see where it is. Do you like this one? Can you chemically modify this one? Or shall we try another one? Shall we modify it here? If so, what should we put? Or should we modify it along this vector? And as we did that, we showed nearly 50 times now that we could get from a millimolar molecule down to a nanomolar molecule within about 6 months. I was pretty surprised that it was so effective. And once we're down at nanomolar, then we could go.
Okay, so basically in fragment-based drug discovery, he grow the ligand. No, I cannot scroll. Okay, he gradually grow the ligand from the small part until bigger and bigger. And he has army of chemists to synthesize all those compounds for him. He starts from a small fragments which are not very tightly bound. And then he improved the binding affinity bits by gradually grow until the whole ligand fit the three-dimensional shape of the pocket. So that's called fragment-based drug discovery. The next concept is pharmacophore. So for the pharmacophore, they categorize the entity in the ligand by using the term hydrogen bond donor, hydrogen bond acceptor, and hydrophobic. So they have hydrophobic part, which is the ring part here, and have hydrogen bond donor, acceptor. So if there is the pocket out of the ligand, so this is the ligand electron clouds, and if the protein is here, if the ligand has hydrogen bond donor, the pocket should have hydrogen bond acceptor and vice versa. So this one, the ligand has hydrogen bond acceptor, the pocket should have hydrogen bond donor. So this is the concept of pharmacophore. Let's come to the case study. So this is serotonin in the pendant that is suitable for the time of Christmas. So people all hope for happiness and satisfaction by serotonin, love and passion by dopamine, and learning, dreaming, and memory by acetylcholine. So it's a nice necklace and nice pendant. And the concept we want to focus is on happiness molecule called serotonin. Depression affects millions of people worldwide. So this is the serotonin molecules that people use for curing depression. Let's see. This is the brain cell, this is the dendrite, and this is the axon. So at the end of the axon, there would be the synapse, which releases this serotonin in red, and serotonin will be reuptake back. And they use the ligand in blue to block the serotonin transporter, and serotonin really gather in the synaptic cleft longer. So by having the serotonin in the synaptic cleft longer, it means that the patients who lack of serotonin become happier again, because these red molecules sit in the synaptic cleft around the region here much longer. The red molecules still here, and then the patient become happy again. So serotonin naturally produced in brain, and in people with depression, it will be in much lower quantity. So once they lack of serotonin, the drug like citalopram will be blocking the serotonin transporter, as I showed in the previous figure. And then the serotonin cannot be reabsorbed back to the nerve cells. And then the nerve, normally they recycle those neurotransmitters. If they don't recycle them, they will be at the junction. And that leads to the increased concentration of serotonin in the synaptic cleft and make us feel happier again. So I download the structure of serotonin transporter. You can see a lot of red helices, and I omit all the side chains, and I show only the citalopram inside the pocket. So if we zoom into the pocket and show van der Waals surface of the cleft, and then show the ball and stick representation of each amino acids, we can see that citalopram is here. And then if they design the citalopram molecule, they would put positive with negative, and then they put the hydrophobic rings together to form pi-pi stacking. So that's roughly how they design drugs. And now we have covered from target identification and validation, and then screening, hits to lead, and lead optimization by docking. And next, we will go to the real case study. Oh, we need to elaborate a little bit more about hit expansion first. So normally the drug molecule should have high affinity toward the, Normally the drug molecule should have high affinity towards the target, which should be less than 1 micromolar. The selectivity versus other targets should be high, and that should have significant efficacy in a cellular assay. So in cell-based assay, it should have high enough selectivity and efficacy. And then the drug likeness as in Lipinski rule of 5 that is in your assigned reading. So that one will mention about molecular weight and lipophilicity and log P, something like that. So it should pass all those criteria in order to become a hit molecule before proceed further into the lead. And affinity, molecular weight, and lipophilicity can be linked to ligand efficiency and lipophilic efficiency. The hit molecule should have low interference with cytochrome P450 enzymes and P-glycoprotein protein. So that means it doesn't cause toxicity or side effects because it interacts with other human enzymes that used to detox those compounds. It should have low cytotoxicity, should have metabolic stability, and also it should have high cell membrane permeability, should have high cell membrane permeability in order to get absorbed into the cell. It should have high water solubility, chemical stability. It should be synthesizable, and it should be able to patent, because if other drug company have patented that molecule before, there is no use to repeat that, and then you cannot, the new drug company cannot claim that as their invention. So there are so many considerations. That's why each drug takes 10 years in order to release for the small molecules drugs. So activity is not enough. Most drugs that fail in clinical trials are active against their intended target, but they fail. So the failure often due to difficulties in administration, drug concentration, drug interactions, or side effects. These are called ADMET issue or absorption, distribution, metabolism, excretion, and toxicity. So you can find the drugs that have perfect three-dimensional shape and charge complementary, but still if they cannot metabolize well, if they cause toxicity, or they cannot be absorbed into the cell, those are all junk. They cannot be used as a drug. And how can the drug be administered should be considered. If you need to inject it every week, that would render it impossible to use as a drug. Normally, oral route is preferred, and it should not be taken too often, such as if the drug is taken 1 time a day, that like about perfect 1 time a day, or once a week should be okay. But if it 7 times a day, that would be impossible, right? So those need to be optimized and put into your drug molecule. And how is it distributed inside the body? If it is the central nervous system compounds, the drug that treat depression, or the drug that treat schizophrenia, it should be able to cross the blood-brain barrier. And how is it metabolized? This can affect drug-drug interaction. And will it cause unacceptable side effects? This one have an example such as in ergot. Ergot is a drug that used to treat migraine, but it causes bad side effects that sometimes lead to leg amputation just by taking the migraine drug. The legs and the limbs, the arms become very dark until black, and they need to like amputate the legs just by having the ergot. It have like 200 other drugs that interact with it, and once it occurs, the patterns can be almost deadly. And applications of computer-aided drug discovery. So these ones you already write to me in the essay, but I will read it for you again. Actually, it's not important because I think you know it by heart by now. If the potential ligand structures are known, docking ligands into the receptor binding sites, or using the structure-based pharmacophores. If no potential ligand structures are known, de novo ligand design can fit the binding site or fragment-based drug discovery. So you have the pocket and you just de novo design or fragment-based drug design to fit the ligand into the pocket. That's imaginary ligand. You grow them from here or you link them from here. When the target structure is unknown, you use homology modeling, which is not advisable because at angstrom levels, you cannot just blind guess. But that's the old concept, and we will know what that can do with AlphaFold. So AlphaFold, they can fold the three-dimensional structure of protein from AI, and they claim it to be very accurate. So I hope that humankind will see more and more from homology modeling and AI fold of the protein, and it can be applicable to the target structures that are unknown but fold computationally, and we can design the drugs to fit into them. And we can use QSAR, linear regression, COMFA, QSA to find the sets of ligand that we know and then predict the target structure from the series of ligand that we have. So Ajarn, we on the drug design, first we need to understand the target. Let's make the analogy. So if the protein pocket look like cat face like this, for example, if the pocket look like that, we can know that, okay, it look like cat. Why it's look like cat? It should have ears like this. So these have ears, eyes, and it should has 3 whiskers and have nose. So understand the target mean what features inside the pockets that we can recognize this one is this protein. And so these are important in order to find suitable mass for fitting with the target enzyme. So if we want to design the ligand in the same way as the mass that's suitable for this target face, cat face, we will have certain things to consider in order to design a perfect mass. So these are a series of pictures that I make analogy. So Tom and Jerry, this one, the structure have low resolution and no colors. So it's also complex because Tom is with Jerry. We cannot design molecule that specific to only Tom face because Jerry is interfere. But if we have the target enzyme that are multi-targeting, we can hit many targets at the same time, as in kinase drug discovery, where we hit so many targets in the same shot. We call magic bullets. That would be a good drug design strategy, so that we design so many, just one drug to hit so many targets at the same time, and we stop cell cycle control. And the Shiny Kitty and Hello Kitty is an example of side effects because they both generate from Sanrio, have the same creator, so they look very similar. And it's hard to find a perfect mass that binds to Hello Kitty but doesn't bind to Shiny Kitty, for example. And then that causes side effects. So it binds to one target and another target the drug cannot recognize and bind another target to, and the system all screw up because it binds to so many things. And Felix and Sylvester is the good example of what a good drug should be. It has a unique feature that still defines its character as a cat, and the mass will be very specific if we want to design a perfect mass for Sylvester and Felix. And in the future, we hope that the drug will be smart drugs, as in Doraemon. So the key to the design: suitable size, shape complementary, and charge complementary. So by screening the size alone, we can discard all the drugs that have like size too large for a particular pocket, and we can screen more than a half just by size alone. And then we can focus into the shape complementary and then optimize the charge complementary. That's the key to the design. So my real life example look like this. So when I do the structure-based analysis, the real life example, I will see the structure in line representation. You can see the ribbon in pink, larger pink like this. And these are all the side chains of many protein superposes on the same pink helices. You can see that this one is methotrexate or the anti-cancer drugs or anti-rheumatoid arthritis drug. So you can see that the drug molecule is very well superposed inside the pocket. So the research question that I have is how to design a better methotrexate, or how to design a better drug molecule if I have given large amount of structures. That time when I did my PhD, the kinase is a very groundbreaking research field. So people are racing for finding the best drugs to attack protein kinase pockets. So I did active site and cleft observation from the whole genome. That is the proteome of the protein kinase. So this is the phylogenetic trees. In the bioinformatics class, we can see it's in the rectangle phylogenetic tree, but this one they create it as a bushy tree, and this is tyrosine kinase, serine AGC kinase, CAMK kinase. So there is classes, and each nodes represent each protein kinase. And the similar thing about protein kinase is every kinase on the map have the same architecture. They looks like this. They have GX, GXX, GX loop in red. So you can see so many G causes so many turns because G introduces flexibility. And there is salt bridge at this DFG loop. Salt bridge means have blue charge character positive, attached with negative charge in red. And the hinge region where the ATP normally binds. So this is the hinge in green. And there is catalytic loop that allows it for the catalytic reaction to occur. And activation loop and DFG loop. DFG loop is here in blue. Sorry for my mistake. So DFG loop is here and activation loop. So all these more than 500 protein kinase in the human genome have one same fold. And another thing I discovered is that the protein kinase all binds to staurosporine with various binding affinity. So on these trees, if the drug binds good, it will have large red circle that shows how well it binds to staurosporine. The staurosporine is the drug molecule for antifungal that looks like this. It mimics the interaction of ATP so well, and there is so much amount of structures in the protein data bank, and it's very rigid. So that's the case study that I start my research career on drug design. So I did that by observing the environment of staurosporine in order to put the staurosporine environment. And then I hope to design a better drugs inside. The package that I wrote during my PhD is called SIMFONEE, where I use 4-dimensional grid to categorize atom in Cartesian coordinate into 4-dimensional grid called X, Y, Z, and type. And then if these 4 characters are satisfied, the grid box are populated by character of 1, like occupancy of 1. So each box, once they have the same X, Y, and Z, and have oxygen of water, I put that box by number 1. If it has nitrogen, sp2 nitrogen, I put the number by 1, something like that. So I can superpose large amount of protein kinase in the database by superpose like 40 of the structures, and I can see the environment surrounding the green molecule. So what I found is this, the conserved atoms and residues. How can I get rid of this? Okay, the conserved atom and residues, you can see that there is certain place inside the pocket. So this one is dihydrofolate reductase. This is folate that mother take. So you can see that there is certain atoms inside the pocket that are found in 100% of time. 100% of the structures that I put will have this atom at this location. And the percent vary, sometimes go down to 30%, but the pocket of the ligand that are used as drugs will be more rigid than the pockets that are used as the natural substrate. And this is again SIMFONEE to observe kinase pocket. You can see that 75% of time, this atom is always here at the angstrom levels. So nanotechnology is 10 to the power of -9 meters. This one is 10 to the power of -10 when we measure. So it's like 10 times smaller than nanotechnology field. So you can see that the ATP pocket have more flexibility than the staurosporine pocket, and staurosporine pocket have induced fit atom at this position. And when I know those, I use those atoms to generate distances. So I basically measure the distance between those frequently occurring atoms and then put in the mileage charge. For example, if you know Chiang Mai, Chonburi, you can know the distance from Chiang Mai to Chonburi by looking at the top of the table like that. And if you know Chonburi to Narathiwat, you can see a number like that. So this is distance matrix generation. And if I put it into Excel table, I can combine the ligand base with structure-based drug design. So this is the binding affinity or the activity that I use to measure how well the ligand could bind. So in the equilibrium, the protein and ligand and protein ligand complex Asst are in the same beaker, for example. So KD or binding affinity is a measure of concentration of proteins times concentration of the ligand divided by concentration of the ligand and protein complex. So what they mean, like if you put drugs and protein inside the beaker, if the drug bind to the ligand, if the drug bind to the protein very well, the concentration of complex will be very large. If the C here is very large, KD is small. So what it tells is that the drug that binds very tight will have small KD numbers. And why dissecting how tightly the drug would bind is important, because drug side effects and therapeutic windows and multi-target drugs can be understood from the promiscuous part or the part that looks the same. And for drug resistance, mutation, and species selectivity, we can look at those from the parts that are different. And from this distance matrix, we can put in table like this. So we put all these numbers inside the Excel table. And in the last column, we put the Y column here from smallest, the best binding KD smallest, to the worst binding, the loosely bound KD. And then if the distance is large, we put it as shade of purple. If the distance is small, we put it as the shade of yellow. So what we can observe from this table is that if we sort the value of y, we can see certain part of the distance x should be large, certain part should be very small. And so we can generate model equation to find if we want the value of y to be very small, this N lobe and C lobe should be closer together, and this part should be expand. And so we can map the structure of Staurosporine environment by lactam binds to the hinge region. The hinge region, more than 75% have atoms that are located at this same location. The indolocarbazole should be expand. And the N lobe and C lobe should be closer together. What we cannot predict at the moment is the type of atoms that have positive or negative charge characters. So we can also generate the tools that observe if there is 2 hydrogen bond, the Ki is small. If there is 1 hydrogen bond, the Ki is moderate. And if there is only 1 hydrogen bond, but instead of binding to the nitrogen, it binds to this oxygen methoxy, the Ki go extremely large or almost doesn't bind at all. And we generate the active site map like that. And then in 2021, just last couple months, I have published a paper called A Machine Learning Platform to Guide Protein and Ligand Design by Anchors and Influential Distance. The server is called MANORAA. So MANORAA has been the most read paper for 5 weeks, and it's just getting more and more attention from the structural biologists around the world by its MANORAA algorithm. MANORAA is a drug design web service. It linked biological big data into genetics, system biology, structural biology, and drug design. The big picture analysis is by linking the ligand to protein, sequence structure, and expression levels. So it can link the ligand to protein, protein to organ and tissues. And that's how we discover the drug side effects. It also allows for linking to genetics data, that is the single nucleotide polymorphisms of the protein. And it also offers the data from biological pathways. That means it shows the KEC pathways. So you can discover the protein in the map of the biological pathways and the reaction of it against several other targets. And for in-depth analysis, the MANORAA allows for protein ligand interaction conservation analysis and drug binding distance correlation function.
MANORAA can help in binding affinity optimization. First, you need to select the template to find the frequently occurring neighboring entities. These atoms will be binned to find the most distinctive parts and measure the distance among them. These distances will be fed into machine learning algorithms, and it will give rise to the influential distances that correlate with Ki, and the drug can be optimized.
Okay, that is the demo video a little bit on the MANORAA.
I will give demonstration on how to use MANORAA. So first you go to the URL manora.org, which will be redirect to here. So manora.org. Then the fastest way, you can click on sample. And then you can see this one is for Staurosporine. And if you want to change to methotrexate, you can put the PDB 3-letter code of the ligand, MTX, like this. And then it will go to methotrexate here. Or you can go to trimethylprim, you can click, put TOP, trimethylprim. So this is top. So it works on any ligand in the PDB with enough structure for superposition. So let's go back to Staurosporine. So this one, you can see that these panels is the Staurosporine, and these are the atoms that will be used for superposition. So the default is heteroatoms, but if you want to superpose more precisely, you can roughly estimate the core structure of the ligand by clicking on structural conservation. In structural conservation, it will superpose all the structure with that ligand on the list of atoms that you input, and the flexibility of the atoms will normally represent by the yellow color, and then it fade to green to cyan to the most superimposable in blue. So it will go from spectrum of yellow, green, cyan, and blue. And you can see that these atoms are very well superposed. And then you can proceed with this list of atoms. And you can go to binding distance correlation. If you click here, it will do the atoms that you select in the previous page, and it will superpose all these atom by atoms. So after superposition, it will give you the list of frequently occurring atoms according to the superposition atoms that you input. And these are the residues that are equivalent. And on the header will be the residues that used for distance measurement, such as this one, the distance will be B11, B12, B20. So it's B11, B20. And then this is the influential distance equation. Normally, the most influential distance will give rise to only one distance. So you will have to manually select the atom for superposition and try to minimize the distance parameters to only one most influential distance. And the green color mean contraction, the yellow color mean expansion. So you can see more clearly in this drug design button. So the list of atoms, this one will be still the same, heteroatoms. But if the ligand are flexible, it should be along the rigid core. Flexibility of ligand will make the superposition very hard to give any meaningful result. So normally it works with rigid core ligand. And then this one gives rise to equation. And if you click on conservation, these are the atoms in the bin that are used for distance measurement. Is the frequently occurring atoms. And these are all the distance measured by these interresidue distances. And if you click on contraction, this is the direction in the pocket that should be contract in order to gain affinity. So this one, the N lobe and C lobe should be closer together in order to get the lower binding affinity. So log Ki very small, that means this distance should be smaller in order to get the log Ki very low. So if this number is small, log Ki is small. And another function would be the pathway discovery and target organs. So this one, if you click on this one, KPCT human, you can see that this is the protein target of Staurosporine. You can click on the pathways. This is autophagy pathways. So it will load into the pathways. And this is the protein that is the target of this Staurosporine. So Staurosporine have many targets. It's very promiscuous, so it gives rise to many pathways and many protein. And if you want to look into the target tissues or organ, you can click on open target. In this open target, if you click to baseline expression, you can see that this autophagy exists in immune organ and in blood and in endocrine gland. And you can also see the affinity. Affinity will be sorted from smallest, which is the tightest binding, to the loosely bound and the non-binding PDB codes. And you can also upload the PDB here.
And this is another server powered by MANORAA. So it's called
InfoCovid.com is a website powered by MANORAA. We provide a unique approach to structural conservation in the pockets. Every atom has a tendency to be at certain place more than the others. We can display the most frequently occurring atoms from the ligands and show them with larger radii in green. We analyze all proteases in the PDB, including the main protease of SARS-CoV-2.